Today, we are looking at DNA plus replication, how it makes a copy of itself. So first we look at the structure. It is known famously as a double helix. And so the helix just means a coil where the radius stays constant. It's often thought of as a twisted ladder. And the rails are a sugar phosphate backbone, and the rungs are the A, T, C's, and G's. And the rungs are the nitrogenous bases, and they're the full words. We have to remember A and T fit together and C and G fit together. And that's very important. Things called hydrogen bonds, if you take Chemistry 11, you'll learn about. They're moderately strong, but they can be broken and reformed. When we look at DNA, we're going to find that we should have the same amount of A as T, and we should have the same amount of C as G, since they fit together. Let's get that out of the way. So now we're going to look at DNA replication. Semi-conservative. What that means is, well, semi means half. So half of the new DNA will be the old stuff, that's been conserved, and half will be new stuff as nucleotides fill in. So the DNA unzips, and nucleotides with bases fill in, A with T, C with G, and that's where we get what are called complementary strands of DNA, where they're half old and half new. So let's look at this diagram and talk about it. So here's some DNA, and it's just unzipped, or starting to unzip. So there's our DNA. It's unzipping. It won't completely unzip, and it will copy sections at a time. So this blue color represents the original DNA. Now, here are nucleotides, and they have the nitrogenous base, and those are the letters, and they're floating around in the nucleus. And if there's a vacant G, a C will pop in. If there's a vacant T, an A will pop in, etc. And the net effect is, over time, when we look at this, here's our original strand from way over on the left, and the red represents the new strand, the other half of the DNA, formed by these nucleotides adding in, essentially locking in position. Now over here, this right side was this strand acting as a template. And these red nucleotides that have filled in are the new strand, or the other complementary strand of DNA. So what we've done is we've doubled up our DNA. We've made a copy or replicated it. On the left, we have this one strand of DNA, half old, half new. And on the right, we have another strand of DNA, half new and half old. And from last year, you should know that's very important. When cells are going to divide, we must make copies of our chromosomes accurately to ensure that we don't run out of genetic information in future cell divisions.